Fungi begin life as microscopic, dust-like spores built to survive harsh conditions. When conditions improve, a spore germinates and produces a hypha, a long, cylindrical multicellular filament. As hyphae multiply and interlace, they create the expansive mycelium. This network forages for nutrients, senses and integrates local cues, maintains continuity via hyphal fusion, and relays signal across the colony. In nature, sometimes extending from a few meters under a log to kilometers across the soil. To deliver these functions on colony scale, each hypha must elongate rapidly and with high spatial precision, driven by a strict program for growth and development. So let's dive just a little bit deeper into the process of fungal growth. In general, filamentous fungi grow in two main stages. First, when a spore encounters favorable conditions, it resumes metabolism, takes up water and nutrients, and swells uniformly in all directions. This type of growth is known as isotropic growth, where iso means equal and tropos way or direction. During this phase, storage compounds are mobilized and the cell wall softens evenly, allowing the spore to enlarge. Next, the cell is ready to develop into prolonged form, hypha. So, at this stage, spore breaks symmetry and commits to one growth site. This is a future tip of the hypha. All the building blocks for the membrane and cell wall are packaged into tiny vesicles and delivered to a single spot on the cell surface. Once enough vesicles accumulate a small protrusion, the germ tube emerges. From this point, growth becomes polarized, meaning surface expansion is confined to a tiny region at the very tip rather than occurring uniformly over the whole cell. A subapical vesicle hub is called the Spitzenkörper. The Spitzenkörper, literally tip body in German, is a dynamic subapical cluster of secretory vesicles and associated machinery that behaves like a vesicle supply center. It directs a continuous flow of vesicles to the hyphal tip, where they merge with the membrane and supply components for cell expansion. Think of it as an airport cargo hub. Vesicles arrive from across the hypha, are organized in the Spitzenkörper, and then are dispatched to the membrane and the hypha tip, where they fuse, delivering wall-synthesizing enzymes and membrane lipids exactly where they are needed. Fascinatingly, fungal cells construct specialized intracellular rods, microtubule tracks, that guide vesicle traffic toward the Spitzenkörper. Vesicles are produced throughout the hypha and move swiftly along these microtubule highways toward the tip. In the final few micrometers, they switch to actin filaments, its actin cables, for short-range delivery and finally fuse with the plasma membrane at the tip. The steep gradient of calcium ions, together with other signaling cues, sustains polarity and ensures the tip grows in a precise and directed manner. The tip of the hypha stays soft and flexible, while the sides are already firm. This difference, together with internal pressure and new wall material added at the tip, pushes the hypha forward. A compact and centrally positioned Spitzenkörper enforces tip dominance, allowing growth to proceed along a single axis. However, under stress or when vesicle dynamics are disrupted, the Spitzenkörper may shift positions or divide into multiple foci, reshaping the colony's morphology. But what about the branching we typically see in mycelial network, the side branches that emerge from the main hypha, breaking its straightforward course? Here, new growth sites emerge along the sides of existing hyphae, a process known as lateral branching. As you might remember, a hypha is a multicellular structure divided into compartments by thin cross walls called septa. The septa act like tiny fences, barriers that still allow cytoplasmic flow through their pores, keeping the hypha connected as one living thread. But because they interrupt the flow a little, they can create spots where vesicles start to accumulate. This is the idea behind the septum as barrier model, which suggests that such regions may serve as starting points for new branches. According to the spontaneous polarization model, vesicles may aggregate spontaneously. Once the cluster reaches a critical size, feedback from the cytoskeleton stabilizes it, initiating a new polarity axis. The growth and organization of fungi are truly one of nature's quiet miracles, an elegant system of coordination, adaptation, and intelligence hidden beneath our feet. The more we learn about them, the more they amaze us. Thank you for watching this video and stay tuned to explore more of the hidden world of fungi.